Hi everyone, good morning and good evening for few others.
Hi.
Good morning, sir.
Good morning, sir.
Good morning,
When I was discussing about the SAS programming, I told you SAS programming is a general programming language. It can be used in an insurance field, it can be used in a healthcare field, it can be used in a banking field. We are here going to implement SAS programming into a healthcare industry, into a pharmaceutical industry. That's why when you learn the concept of a CDISC or when you learn the SDTM, ADAM, and TLF, we call it as a clinical SAS. I hope you guys are ready. Only prerequisite required for this training is that you should be good enough or you should be good with your programming skills, which is base SAS. The only prerequisite is required for this. We have few new members. We have Pilpa, Krishna, and Ishtani. All of you have joined today. I can see.
Yes, sir.
Okay.
Yes.
So we have three members who are already aware of a SAS programming. So they wanted to start with the clinical SAS. So from your end, only thing, as I said, you required is a knowledge of a SAS and a interest and willingness to learn. That's all. Hear me carefully, and I will make sure that entire clinical SAS concepts would be very easy for you to understand. I'll make it very simplified. I'll not use any kind of a unnecessary difficult term for you to understand. I'll make sure that whatever terms is getting used in industry, same terms I will use, but I'll make it more simplified as well for you. On the assignment, as I say always, make a notes later on when you go through a recorded sessions after the training, and you will get all this concept very easily. And actually, clinical SAS in the entire concept of a CDISC, which is STTM, ADAM, and TLF, is very interesting to learn. So if you take an interest, and I will try to create some stories and try to make you explain. And you try to relate with a real-time examples. So that way it would be easier for you to learn the concepts of a clinical SAS. So what we are going to cover is STTM first, and then after we will be having a mock interview. Once we complete STTM, mock interview, then we'll start with ADAM, then again mock interview, TLF, then after mock interview, and we are going to also cover about SDRG, ADRG, define. These are some documentation which is required along with what you are creating, along with, let's say, you are creating some data sets, you are creating some reports. So along with those data set and those reports, you need to also create some documentation that is called as the SDRG, ADRG, or define. We'll see all this thing in a detailed manner. We are also going to cover a basics of R at least so that you can create a listing. And the purpose for keeping this session is to just make you stand out, because everyone will go ahead with the knowledge of a SAS. But if you keep in your resume that you know a basics of R, so you will be having a little more advantage. Annotation and specification writing. And at the end, we are going to have a how you can prepare your resume or CV and how we should market it. So this is the overall plan we have for this training. Okay, so let's start with trying to understand where we are working. As I said we are going to use the SAS programming into a healthcare industry, pharmaceutical industry. What kind of a data we are going to receive? Going to receive a clinical trial data. What is the meaning of clinical trial? Many of you would be knowing about it. So once the scientists have the combination of a
Before, so can I say that
yes, go ahead.
uh, after completion of in vitro studies, the drug goes to the clinical trials process on volunteers, respected regulatory market, for taking the safety and efficacy for respective molecule.
Correct.
After getting that data, then it goes to the
Yes. So we are having a clinical trial. When we go for a clinical trial, once a scientist have the combination of a compound or formula after the years of a drug discovery process, then they go for a clinical trial. What is the major reason or what is the purpose of a clinical trial? Is to find out the safety of a drug. how safe the drug is. Efficacy, how effective the drug is. Dose tolerability, what should be the route of the administration, what is the PK parameters. These are the main aspects behind the clinical trial. This sponsors company, they filed a IND, investigational new drug, and then they go for a clinical trial. So we have several phases in a clinical trial. First of all, we have a preclinical. Before we try on a human, before the drug is tested out on a human, it would be tested out on an animal, which is called as a guinea pig. So if everything is good in a preclinical phase, then we start the trial on a human. Starting with the phase 1, then we go ahead with the phase 2, and then phase 3. What's the difference in all these phases? Phase 1 is little shorter in length. This is not the standard definition, but it remains kind of similar to this, what is mentioned here. Phase 1, it remains for 1 month. It also goes for sometime 2 months, 3 months. Duration is 1 month. Number of people would be less. And main reason is to know the safety of a drug, how safe the drug is. That is the main reason. Purpose, how does the body process the drug? What are the side effects? This is the reason behind, these are the purpose behind the phase 1 clinical trial. Major purpose is safety. We are not looking for the efficacy here. We are majorly focusing on the safety of a drug. Is it safe for a participant or not? How effective the drug is. If let's say there are a lot of side effects happening, so then again that trial would be terminated. Phase 1 mostly for a safety purpose. Then we go ahead with the phase 2, just that the duration is little more lengthier now. We have few more number of a subject, and safety is constant. Will not compromise with safety anywhere. We always look for a safety. And here we are looking for the efficacy, how well a drug is working. How much should be the dose for that particular phase 3? You have a duration is from 6 to 12 months. Some trial also goes for up to 3 years, up to 2 years in a phase 3. More number of a participant, safety, efficacy. And we also see if whatever the risk is there, whatever the side effect is coming up, is it something we can compromise with what the efficacy, what the effectiveness is coming up? Does the benefit outweigh the risk? And if all these trials are successful, it's not like all the trial will go ahead parallelly. Once phase 1 is completed, if we see a potential in a drug, then phase 2 would start. After phase 2 completion, then phase 3 would start. If we see a potential in a data, if we have a effectiveness, even if there is no effectiveness, if drug is not safe, we still have to follow the process of submitting all the data document to a regulatory authorities, even for a terminated study. So we have a regulatory authorities. So in US, you have a FDA. And most of the time, you will be working on a FDA approval or FDA regulatory authorities. Your drug will go for a submission to a FDA. If it's in a Japan, we have a PMDA. If it's in a European country, you have a EMEA. For India, you have a DCGI, Drug Control General of India. But mostly, I have in my experience, I have seen 85% of the study would go for a submission to FDA. Application submitted, and then after it will be reviewed, it will take again so much of a time, and then it would be approved. When it is approved, then the drug is available to a public. And then comes the post-marketing phase. Mostly we get a data for phase 1, phase 2, phase 3. We do the analysis of the data for these phases, not for the phase 4, not for the pre-clinical. The drug is in a market, again, they will keep on seeing if it's safe or not, any kind of a side effect is happening, if the cost effectiveness is there or not compared to other drug. So company, the sponsor will keep an eye on all these things. So even if you just turn the packet of any drug, you will see some number would be there, right? Customer care number. email ID. If you have any issues, anything, you can reach out to them and they will get the feedback about it. That data, the company, again, when we work inside our company, we won't get this phase 4 data. There would be a small team for a phase 4, not in a company, they will keep it separate. Mostly when we work in a company, we won't get a phase 4 data, but they will receive the data somewhere. Any question with this entire thing? So you should know which kind of a data you are getting before we look into the SDTM. What is the meaning of a randomization? So let's say here you have 6 participants or 6 subject. First 3 subject would take, let's say, drug A, and rest 3 will take drug B. Here we are assigning a subject to a treatment. That is called as the randomization. And this will not be something like a investigator will choose or a sponsor will choose that this 3 person has to take drug A. It is by the virtue of a luck. You can just take a think example of like how we do a toss before playing some matches, cricket matches, right? It's all by the virtue of a luck we decide who will bat, who will ball first. Same way, to avoid the biasness. There should not be any biasness. By the virtue of a luck, there's some process by which randomly they will pick that these subject has to assign to a drug A treatment and these one to a drug B. It's a ratio. It is a ratio decided that we have a ratio of 1 to 1 of a randomization. That means let's say we have a 6 subject in our trial, 3 would be randomized to drug A and the remaining 3 would be randomized to drug B, just to avoid the biasness. When you see this, let's say here you have these all 3 subject will take a drug A and these 3 will take a drug B. We call a group of a subject as arm, treatment arm. Arm means, definition of arm is a group of a subject who are or who is supposed to receive a same kind of a treatment. If I am assigned to a drug A, I will only take drug A if the study is parallel. Arm is, this is very important. Definition of arm is a group of a subject who are supposed to take a same kind of a drug, not similar, same kind of a drug. Drug A, if the subject is randomized to drug A, treatment arm, they will only take drug A throughout the course of a trial. In the case of a parallel study. Purpose of a randomization to avoid the biasness. Randomly assign the subject to a treatment group. Treatment group is also called as a arm. So when I say arm or when I say treatment group, both are same. Till now, is it clear? Any question? Do feel free to ask me. Let's understand the process flow of a clinical trial data. Somewhere clinical trial is happening. This is the process flow of a clinical trial data. So clinical trial is happening, how a data would flow. Not that clinical trial is happening and we will directly get the data for our data analysis. When we work in a SDTM or in ADAM or in a TLF, there is a certain process flow through which data would come. Clinical trial is happening, data would come through a EDC tool. EDC means electronic data capturing. In earlier time, there used to be a paper CRF kind of a thing, where the data would come in a paper and then it would be again entered through the process of data entry to a system. Now we have an electronic data capturing tool. These are some of the tool you have. Rave, Inform, Oracle, and most commonly nowadays, many companies using Viva Vault. The trial which I'm working currently, it is using Viva Vault. Through these EDC tool, the data would come to a team called as clinical data management. Or we also call this as a DM, data management simply. We are not working here in a CDM. Once data is coming to a CDM team, who will enter data? Someone from the site Site means where the trial is happening. It may be a hospital. We won't call it as a hospital. We'll call it as a site where trial is happening. Some site coordinator will enter all the data into this tool and it will come to a team called as CDM. What CDM will do, they will give you a SAS datasets. Before that, they will do some edit check just to see if the data is clean or not. They'll check the data has the quality or not. They will do some medical coding. They will also do some reconciliation kind of a thing. They will also do some adverse event kind of some listing they will create. That is different. That is a CDM job. What we should understand is we are going to receive our data, or we call it as a raw data, what we are going to receive, input data set for us. That we are going to receive it from the CDM, Clinical Data Management Team. From this tool, they will create a SAS data set because we are our system are made to use a SAS data set, right? So they will create a SAS data sets and then they will provide to our team, which is called as the SDTM. Here we are looking into in this team. From here, our work start as a statistical programmer. If we receive a data, what SDTM team will do? We will make a data set standard. Data what we receive is not a standard from the CDM. When we say data has to be standard, it means we have a standard data set name, a structured or standard variable. We have something called as the SDTM implementation guide. From that guide, we have to follow all the rules and regulation, and we have to make this data set, which is not a structure, which is not a standard. We will make it standard and structured. Every bit of a data which is collected in a trial, it should be easily identified. That is what we are doing in SDTM team, making a data set standard. If the data set is not a standard, it would be very difficult for the regulatory authorities to review data. And if it's taking more time, let's say all the sponsors, who are the sponsors? We can take example. Let's say you have Novo Nordisk, you have Novartis, GSK, Pfizer. These are the some of the common. If we have a data set already standard, it will take less time for the regulatory authorities like FDA for a review. And if we are able to save the time, that means we would also save some investment as well. And data set is more consistent and structured. Understood why we have SDTM? That's the whole purpose, making a data set standard. Once the data set is made a standard, it would be passed to a next team, which is a ADAM. We are creating a SDTM data sets here using the raw data set what we have received from CDM team. Once SDTM is completed, we are going to create a ADAM dataset. Purpose of our ADAM dataset is to create a dataset ready for the analysis. We are not doing any analysis here. We are just making a dataset standard here using the guidelines of a SDTM. All the analysis we are going to perform at the ADAM dataset level. And once that is complete, ADAM dataset would be the input for your TLF. Using all the ADAM dataset, we are going to create table, listing, figure, or also we call it as a graph. We cannot create TLF just by using the SDTM dataset. It has to follow the process sequentially. Once your TLF is created, then our study submission package is ready. We're going to submit all these things, SDTM datasets, ADAM datasets, TLF, along with the documentation to the regulatory authorities for their review.
I have a question.
Yeah.
You said that while implementing the data for SDTM, we don't do analysis on data. But I'm just curious, as far as I remember, we do a lot of analysis on data. I don't get this ADAM part.
Come again.
So you said that? while we are performing SDTM, while we are just making the data standard, we do a lot of analysis on the data, right?
Correct.
We perform a lot
Not in
of
SDTM.
checks.
SDTM purpose is to just make a data set standard as per the SDTM implementation guide. We are
Right.
not doing any analysis. For analysis, you have a separate document, which is called as SAP. We'll look into that when we start Adam classes. Using that document, what is the requirement is there for analysis? We'll go through the entire document and then we'll decide how analysis has to be performed at the Adam level.
Sir, this one for STDM analysis, so I could understand from the chart flow. From DM currently, if data is transferred on a monthly basis, as the workflow happens on a monthly basis, like we receive from the data from the DM, again we do the data set analysis, standard data set, then
Yeah,
add them like that
yeah.
every month the cycle will happen.
Yeah, so this is something which I'm going to discuss. So yeah, see, clinical trial is happening. Let's say the duration is 6 months. And it is decided with the collaboration with the DM, STTM, everyone. We have agreed that we are going to receive a data, let's say every month. That means we need to create this data set every month. Whenever we receive a data, we are going to create our data set. First time, let's say that it is planned in the next month that we are going to receive a data for a first time. That time you have to write the code from the scratch.
Okay, sir.
And again, when the new data keep on coming, it's just a rerun of your own program. That's all.
Okay.
Okay. So yeah, it's all depend upon the frequency of a data transfer. If it's the trial is for, let's say, for 3-year duration, which is of a phase 3 trial, and it is decided that every 3 months or every quarterly you are going to receive every 3 months. That means every 3 months you need to rerun your data set. Entire process will start. Only thing is this submission will not be done until unless you have the trial completed. This we don't do every month or every 3 months. Submitting the package. It's only when the clinical trial is completed, then we are going to submit everything at the very end, after the end of 3 years. Before that, every 3 months, this data set has to be created. Again, Adam data set has to be created. TLF will be created. Why it will be created? To just know that, see, the trial is ongoing. That means we have more and more data coming on. More and more subject is getting recruited. So we will get to know if we have more data, more subject, we'll get to know how the safety is, how the efficacy is going on. So it helps the clinician, safety team, right? These teams to understand how the drug is working. At the end, it will go for a submission. Okay, now we are going into the SDTM. Now we understood what kind of a data we are receiving, everything. We have got the overall view. We'll go more specific now into the SDTM. We know why we have a SDTM standard. But let's see what kind of activity we have in a SDTM. First of all, you have a protocol. Protocol is one of the document which is created by a clinician, statistician, safety team would create protocol. You don't have to do anything. When you start your work in SDTM, you are going to have this document protocol. What is the meaning of a protocol? Protocol will say how a trial is happening, when it is going to happen, why the trial is happening, where it is happening. All this how, when, why, where, all these answers to all these questions would be given in a protocol. What is the objective of a trial is, what is the end point of a trial is, what is the study design, what is the treatment we have, what is the criteria we have, all these things would be mentioned in the protocol. I'll share the document with you after the class. From this protocol, we are not going to do anything with the protocol. I mean, we don't create this. We have this document in our place. From this protocol, our activities start. We create a CSV file or Excel file, which is called as the TDM, Trial Design Model Excel file. What is this TDM? I'll go in a detailed manner in the next classes. Just you understand for now, this is one of the documents. The TDM is a one Excel file which SDTM team has to create by referring to the protocol and nothing else. Only by referring protocol, we are going to create TDM, trial design model documents. Once you have
Sir,
TDM,
what is the format you said? I just missed it.
Excel or CSV file.
Okay, thank you.
Once you have a TDM, then SDTM team would create or they would annotate the CRF. What is the meaning of CRF? CRF stands for case report form. If you look into the CRF, I'll show you all those things. This is called as a CRF, case report form. By looking into the case CRF, case report form, we can get to know basically what kind of a data we are getting, what kind of a data is getting collected. So if you see here, I'll just show you one of the simple page for now. So you will get the date of birth of a subject. What is their race? What is their sex? So these are the information which you are going to get in your raw data set. See, when I say here EDC tool, right? I was discussing about EDC tool here, right? Rave Inform Oracle. From this tool itself, CRF is created. Tool only, the CRF is created. And this tool would say what kind of a data we are capturing in the study. That same data we are going to receive when we work on a SDTM dataset creations. Nothing else would come up. All the data what we are going to receive, it is mentioned in the CRF. So we are going to do a annotation of the CRF. What I have showed you, it was the ACRF. There is something called as blank CRF. Initially, when we receive a CRF from the DM team, it would be a blank CRF. When I say blank, it means, you see here, these all things are called as the annotation. Whatever you are seeing in a blue color, it is called as the annotation. We are going to receive a blank CRF. Blank doesn't mean that it will be entirely blank. Only whatever she's seeing in a black color, these informations we will get. We have to annotate the CRF. What is this annotation? We'll look into this later. This is one of the activity in SDTM. We need to annotate the CRF. Once this is annotated, we consider this as the ACRF, annotated CRF.
Sir, please quick question, sir. Sir, these annotations are done manually or do we have any system in place?
There would be a tool to annotate that. That is basically a PDF editor. So some company uses Acrob only, some company will use Nuance PDF. So it depends on the tool. Basically, it's a PDF editor tool. Okay.
Sir.
After your annotation of the CRF is done, then there's something called as a specification writing. What is a specification? It gives you an idea about what are the data set we are going to create and what logic we need to use. We have a data set, list of a data set to be created. And also in each data set, we have list of a variable and the logic of it. So if I show you, this is something called as SDTM. This is something called as the SDTM specification. You can see these are the list of a data set, or we also call this as the domain. So if you open one of the domain, let's say I'm opening DM or anything. Let's say I'm opening AE dataset or AE domain. These are the list of a variable which we have to create. These are the label I have to assign. These are the logic I have to use. It gives you idea about the data set and the variable, what we are going to keep, and the logic which has to be used. This has to be created by a SDTM team. Once that is created, to create this specification, we need to refer. See, for annotation of the CRF, we are referring TDM, and we also refer SDTM implementation guide. I'll show you what is this implementation guide. We also check the raw data as well. And there is something called as CT is not required for the ACRF. When we do a specification writing, that time we are referring protocol, we are referring TDM, we are referring ACRF, and we are referring all these things. SDTM implementation guide, raw data, what we have received from the DM team, and also there's something called as a control term document. I'll show you all this later. By referring to all these things, we write a specification. To create TDM only, we are referring to a protocol. To annotate the CRF, we are only referring to the TDM. And we also refer to the SDTM implementation guide. Once the specification is done, then we create a data set by writing a program. Till now, everything was like kind of a manual process, writing some document. Excel file, this is a PDF editor file, and PDF file, this one is. And once these all things are in place, you have TDM, you have ACRF, you have a specification writing, and these things would be there, right? When we receive a data first time from the DM team, we will receive SDTM raw data sets. And IG would be there, control term. These are the fixed document which we use for all the studies. So these would be there in the SDTM team. So we write the program, SAS program, and we create the SDTM data set by referring to all the thing. By referring to all these documents, everything, we create a data set or a domain called as a something. We call it as a domain. So we create a domain here, data set here. First data set what we create is a DM, which is called as a demographic. Once demographic is completed, let's say, then we work on the SDTM datasets, all other. First of all, we create this DM. Why we create DM? Because DM dataset would be the input. Along with the raw dataset, what we are receiving from the DM team, we also need to use the demographic domain if I'm working on another dataset in SDTM. That's why we create this dataset first. Once this is in place, then we go ahead with the all other datasets in SDTM. Whenever you create a dataset, you will refer all these things. Once the dataset is created, it would be validated, or also we can call it as a QC. What is the meaning of a QC of a dataset? It would be checked. So there is something called as a double programming. Each dataset or each domain would be done by two person or two resources. Let's say you are working on the production side, I am working from the QC side for the same domain. And at last we will compare. See, our logic should be same, right? Let's say I'm creating some variable. Ultimately, the output should be, data set should be same, right? Only thing is how I'm writing a code would be different. My mindset is different, your mindset is different. But finally, it should match. That is called as a validation. So it would be all the data set, what is created by a production programmer would be validated by a QC or by a validated programmer. So everything is matching, then only we call it as a data set is QC pass. Once that is done, then we call something else. There is some tool which is called as a Pinnacle 21, or also in a short we call it as a P21. In a P21, we will just check whether our data sets, what we have created, it is following the guidelines of a SDTM or not. What is the guidelines? This is the guidelines which is given by a CDISC team, SDTM IZ. Whether the data set is following the SDTM implementation guide, rules and regulations or not. That is what we check in a P21, in a Pinnacle 21. How we run this, all this thing we'll see in a few further classes. Once Pinnacle 21, everything is clean, everything is passed, then you have your SDTM data sets. You have to create a XPT as well. You have the specification, you have the ACRF, you have TDM, you have P21 report, which comes from this tool. You have a two document which is called as the STRG and define. The data reviewer guide and define. These all things would be a part of a SDTM team. These are the final document and data set what we have. This would be the part of a submission, or if let's say the study is not going for a submission. We discussed that every 3 months we are receiving a data. So these all document data set would be the input for the next team, right? They will again use for their analysis by the Adam team.
Okay.
This is the overall process you have, overall activities you have in the SDTM. These are the final output you have. This is what you have to create: data set, spec, CRF, TDM, all these things.
Sir, like what are the like DM data first we get, and in QC part, like which is the like how we'll compare with the, like which is the first data set, and like
DM data set you will compare first by using proc compare.
With the QC, like with who, which data set, like
QC data set will compare with the production data set. Person is working on a same data set. Let's say I am working from the QC side for the same DM domain. Let's say you work from the production side. I will be comparing my data set with your data set. SDTM is one of the team. I was discussing about this, right? So overall, this ADAM and TLF, in most of the company, ADAM and TLF would be in a same team, commonly termed as biostatistics. You may get a chance to only work on SDTM, or some companies hiring, let's say, for end-to-end. Then you will get a chance to work on SDTM, ADAM, and TLF. If you're working for a sponsor company, they have a separate team mostly. Even in a CRO, like you have IQVIAs, LabCorp, Seniors Health, all those companies have a, may ask you to work on, I mean, they only hire you for SDTM, or they may only hire you for the biostatistic team. So it depends. And sometimes they also hire for the end-to-end activities. Once you know SAS program, you know the guidelines of SDTM, ADAM, you can work anywhere. So it all depends upon the requirement, which company have requirement for which team. You may only go for SDTM, you may only go for a ADAM TLF. You may also get a chance to work on everything end-to-end. So only thing is, when you know everything, you have a more chances of like going to, I mean, the company, when you're seeing some requirement, you're fulfilling more of the requirement. Even you only know STTM, again, you are seeing a requirement for the ADAM TLF team, you won't be eligible there. So it's good to know everything. And in the beginning, don't be a choosy. Go for whatever team is, I mean, whatever they are hiring for. When they are hiring for only for STTM or only for this one, go for it. And there's nothing as like a freshers or as an experience, you will get a chance even as a freshers to be hired as STTM in a ADAM TLF as well. SDTM team and ADAM team. Once the data set is QC pass, everything is matching, then we need to run the P21 on both SDTM datasets and ADAM datasets to see if it is following the implementation guide. How we have a SDTM implementation guide. Similarly, we have a ADAM implementation guide as well. Okay, let me show you the implementation guide, called as the implementation guide. And latest version is 3.3. And there are few changes, few of the new domains are added in a 3.3. That's the only change. Other than that, everything is all same. This implementation guide is given by a CDISC team. CDISC is the team who have given this SDTM implementation guide as well as the ADAM implementation guide document. CDISC Submission Data Standard team. This is the team who create this implementation guide. Whatever we do in SDTM, it's as per this document. We don't apply any extra brain from our end. Only thing is we follow this document. Nothing apart from this. Nothing will assume. We just have to follow this document. So when you work on SDTM, this is your, I call it as when I learn. So what I was told is that this is kind of a Quran, Gita, Bible, everything for the SDTM programmer. SDTM, what does SDTM stands for? Study Data Tabulation Model. I'll talk about first domain. Before that, there is something called as SDTM standard domain model. So you have different classes in a SDTM. When I say classes, so let's say first I'm looking at special purpose domain. This is one of the class. Then after I have an intervention, I have an event, I have a finding. These are my classes. Finding about trial design domain. This is one of the class. I was talking about that document, right? TDM. We need to create some data set as well. That is one of the class. Relationship data set. These are the classes we have, or classification of the data set we have in a SDTM. Under that, you can see the list of your domain or list of a data set. Special purpose, you have, let's say you are seeing a demographic. What is demographic? You have a subject level information. Let's say what is the subject ID they have when they took a treatment for a first time, what is the country they belong to, what is their race, ethnicity, sex, everything. These are the subject level information. Those all data will be kept in a one domain called as demographic. When they have come into a trial, when they have visited a trial, that is all again in a one domain. Like that, you have all the different sets of a domain. We are starting with our special purpose domain, and we'll start with a DM first. What is all intervention event? We'll see all these things. Definition of a domain is, as you know, group of observation which shares a common interest. When I talk about demographic, it is all about the subject level information. So we are sharing common topic is here is subject. That's the definition of a domain. We are getting a same or similar kind of a data in one of the domain. Let's say I have in a trial, some subjects are suffering, getting some side effects. For that, we have a different data set called as adverse event. We'll not keep everything all combined. As per the standard, we have a different data set for different topic. I'll just give you introduction of a DM. Yes, so as I said DM is the first domain which has to be created. It describes each subject in a clinical study. This is a data set which have the subject level information, as I said and this is a parent domain for all other data sets. I talked about this when we work on other data set, we need to have a demographic data set already created. That's why we consider this as the parent data set. I'll come up to all these things, but before that, I'll go through the example here. You have a list of a variable. All these variables are listed here. These are all the standard. This is first a standard data set. You have a standard variable as per this implementation guide. And you have a label for it. Label is basically short description, right? We know how to assign label in a SAS. For each variables, you have the label as well. And you have a type, you have a control term, role, all this thing we'll see. We have a note as well, some description about this variable. So I'll talk about that later, but let's talk about this example first. This example itself, those all variable would be clear to you. What is the meaning of it? And we'll talk about then after, what is the role, what is the control term, and what is the core later on. See example here, this is a study ID. For each clinical trial, you have a study number, study identifier. You have the first variable study ID. It would remain same throughout the study. In all the domains, in all the data set, it would remain same because for one study, you have one unique number. Domain. This is all about the demographic domain, right? So the short name is DM. If I'm working on adverse event data, domain would be AE. Unique subject identifier and subject identifier. For each subject, you have a subject number assigned, subject identifier. See, all the variable has some meaning. It's not like you have to by heart remember this variable randomly. You have to just see the meaning. SUBJ stands for subject. ID stands for identifier. U stands for unique subject identifier. All the variable has some meaning. So if you remember with the meaning, it is easier for you to remember and also to, like, whenever somebody asks you, you can easily recall it. Subject ID, you have the subject number assigned and you have a unique subject identifier. What is the meaning of this? Why we need two numbers for same subject? What happens is there might be one subject who may participate in several trials. For one compound, there may be sometime multiple studies going on. And that subject is participated, let's say, in a 2 trial. This unique subject identifier will make it unique within the study. This subject identifier is unique throughout the compound. I have participated in 2 trials. My number would be 001 in this study as well as in another study as well. And I have the unique number assigned in this study. So this is the unique number which I have for this particular study. Again, in a different study, I will be having a different user ID. It is basically a combination of or concatenation of a study ID, your site ID, and subject ID. Once you concatenate these three variables, site ID is basically where a trial is happening. Once you concatenate, you have a unique subject identifier within a study. RF STDTC. This is the date when subject took a treatment for a first time. Reference start, date and time. C stands for whenever you have a date, this is a character variable. And also it's a date. So that's why you have a C here at the end. RF STDTC, if you read a label for this, subject reference start date and time. It basically stands for when a subject took a treatment for a first time. When a subject took a treatment for a last time. RF ENDTC. Subject reference end date and time. And next variable was RFX STDTC, date and time of first study treatment. And RFX ENDTC, it stands for date and time of a last study treatment. So what's the difference actually? Both, when I define both the variable RF STDTC and RFX STDTC, both are the date when subject took a treatment for a first time. When it is different, whenever you see this X is there in between RF STDTC, that means when a subject took a treatment or when a subject took a treatment for a first time for a protocol specified drug. So let's say example of an oncology study. In an oncology study, before subject take a actual study drug, they will go for a chemotherapy. That chemotherapy treatment date would be your RF STTTC. That is the date. I mean, we need to see when they took the chemotherapy for the first time. That is your RF STTTC. And when subject took the actual oncology drug, or that is also the protocol specified drug, that date is your RFX STTTC. That's the difference. For all other studies, let's say the studies for neurological, for cardiovascular, anything, value for both RF STDTC and RFX STDTC would remain same. You can see in this example itself, the value is same because this is not the example from the oncology trial. How to define this, all this we'll see programmatically tomorrow.
Sir, sorry to interrupt you.
Mm-hmm.
Sir, that RF XSTTC you said, right, sir? So that is a study treatment of that is study treatment, right? Which is nothing but EX domain.
Protocol specified, yeah.
EX domain, right, sir?
Leave the EX and all. Just understand protocol specified treatment. Actual study drug. And if any supporting drug, in example of oncology, I have told you, right? On chemotherapy subject will go for. That date would be your RF STDTC. Other than that, for all other study, value would be same.
Got it, sir. Thank you, sir.
RF ENDTC, as I said you, same thing. In an oncology study, again, when subject took a treatment for a last time, the actual study treatment for a last time, that is your RF ENDTC. And you have a RF ENDTC when subject took a treatment for a last time. So whenever you have X in between, that is all about the protocol specified actual study treatment. First time, last time. ENDTC means last time, end date. RFICD, this is a spelling mistake here. This is the RFICDTC. Reference start, IC for informed consent, DT for date. And C is for, it's a date in a type character. When a subject signed informed consent, only one time they will sign. That date is mentioned here. Participation end date. Let's say the study, one of the subject has completed the entire set of a follow-up. First of all, they went for a screening. Then they went for the entire treatment. Then they went for a follow-up. Let's say they completed the follow-up. So that when they have completed the follow-up for the last time, or whenever the subject was approached for the last time in a trial, that is your participation end date. let's say one of the study in one of the let's say one of the subject, they discontinued in between the treatment. That date, whenever they were contacted for the last time, whenever they were contacted for the last time, that is your RF participation ended. It's not only that, only if the subject is completing the follow-up, that date is only the participation ended. Whenever they end their participation, it can be in either of the phases, in a screening phase, in a treatment phase, it can be in a follow-up. Whenever they end their participation due to whatsoever reason, that is your participation ended. Or let's say they have completed everything, then in that case, your follow-up date is the participation end date. Site ID is where a trial is happening. So for each site, you have the unique number, site identifier. Investigator name, INV for investigator, NAM for name. Birth date, since again this is a date and you have a character variable. One thing to notice, if you notice here in SDTM, all the date variable is in a character type. And what is this format?
Yes, 8601.
Yeah, ISO format. So as per the SDTM, your date has to be character and it has to be in a ISO format as per the SDTM implementation guide rules in all the study. You have an age of a subject, you have an age unit, you have a sex, you have a race, you have ethnic. These are the different set of information of a subject. ARM CD we know already, and ARM. So ARM is basically a group of a subject who are supposed to receive a same kind of a treatment. So this subject 001 is randomized to drug A treatment arm. That means they will only take drug A. This is an example of a parallel study. This 001 only will take drug A. 002 subject will take placebo. One of the subject number 4, if you see here, treatment start date, everything is missing. The reason is they failed the screening test. That's why the arm is screen failure here. ARM code, this is CD for coded term. ARM code this is. So for drug A, the coded term is A. For placebo, coded term is P here. So one-to-one mapping is there in between these two variables. The coded term, a short value for the ARM variable. If you see, whenever you see placebo, it would be P everywhere. If it's drug A, then A. So one-to-one mapping is there. It's not like somewhere A, somewhere something else. It should be having a same kind of a name. Talk about this, and you have a country. Country is basically where a trial is happening. In which country? It's happening in the US. And you have two different sites selected for it. I can see 001 and 2. It's happening in the USA. And you have an actual arm. So what is the difference between this arm and this arm? We have an arm we know already, but we also have ACT. ACT stands for actual arm. That means when I was discussing about arm, the definition was who are supposed to receive a certain drug, right? A group of a subject who is supposed to receive a drug A. But whether they have taken drug A or not, we can get to know from the act arm variable. So this is the planned arm we have. This is the actual arm we have here. After the randomization, this subject was planned that they will take drug A. But whether they have taken drug A or not, you have in an actual arm here. Placebo was planned after randomization and they actually took the placebo. If let's say plan 1 is drug A and due to some mistake, they took, let's say, placebo, then the value would be placebo here in actual arm. And you have actual arm code. Same thing. You have a coded term for the act arm. Understood all this example? Any question in this? How to create this variable? We'll see this tomorrow when we write a code for this.
Are there any information about the full forms of these variables in this PDF?
Full form in the?
In this PDF you're looking?
Yeah. You can see these are all the full form. So whatever I said you, study ID, study identifier, it's mentioned here. It's a label.
Okay.
And if you again want to read about this unique identifier for a study, same thing I've explained you at the down. When I talk about use subject ID, you can see here concatenation of a study ID, site ID, use subject ID. It's a unique subject identifier. You can see what is the type of the variable here. Type is, these all variables are character. We'll see all this thing. See, these are the date variables. Format is mentioned in ISO format. Okay, so everything is mentioned in the IG.
And sir, so in this demographics domain model, these are only the variables present, or are there any other variables too in this domain?
No, everything has to be come up from this list. Nothing we can add extra, as SDTM is all about making a data set a standard as per these guidelines. So nothing else or extra we will add. If I'm working on DM, all the variables should be from this only. And it's not like all the variable is required. I will talk about how to select a variable. So you are seeing a list of a variable, but not all the variables are required. Let's say in your trial, as I told you, a CRF is there, right? So let's say they are not collecting the information about the investigator ID, or let's say investigator name. That means that these two variables are not required. We will not keep these two variables. But all the variables listed in the SDTM specifications should have the name here. It should be listed here. Nothing extra we can add. If we have to add something, let's say some information is there, which is extra in the CRF, and we don't have a suitable variable here, then there is some other process what we have to follow. But when I open a data set finally, I should have only these variables. Nothing extra. If I have extra variable, then P21 will throw error. Any other question? I know the sessions is little lengthier today because this is the first session. So it takes some time. From tomorrow, we'll try to keep it. I'm not giving any assignment for today class. I have a plan. I mean, how we work on this assignment is I have a list of for SAS, I can see. These are the names we have. So once you work on the DM and sub DM domain, then you can send me the assignment. I will check your program. I'll check your data set. And then I'll keep you accountable through this tracker. So from today's class, you don't have any assignment, but the thing is you have to make a note. A lot of information I talk about, a lot of things I talk about, right? So if you don't make a notes, I'm telling you, you'll easily forget all those things. All these PPT, which I am talking about, these all things, you have to make a note of everything. How I define a variable, you can make a note of it. Otherwise, you'll forget about it. There's no chance that you will remember because you are hearing all these things for the first time. So you may have to go through these sessions again. Go through a recorded sessions once again after, I mean, it will take half an hour for these sessions to be again available for a recording. So you have the recorded sessions entirely. And you have a lifetime access for all these recorded sessions of all these live classes. So that time you can just pause it and you make a note. Don't make any note during the classes. Make a note, work on the assignment. That's the only mantra I have for you guys. All good? Anything? Any other concern?
Okay, thank you.
Okay, thank you. Let's meet again tomorrow. Bye, everyone.
Thank you.
Thank you. Bye.
Thank you. Bye.